Hello, I'm Trond.
And I'm John, and we would like to welcome you to this immersive experience inside the human body, showing the journey of drugs from the moment they are swallowed and until they are distributed throughout the body.
Throughout this journey, we will explore drug absorption, which is the process by which drugs enter the body when taken in tablet form. In the throat, the tablet is swallowed and descends into the esophagus. In some cases, larger tablets may get stuck in the esophagus of patients with a narrowed lumen.
However, for the majority of patients, this stage of transportation is uncomplicated, and the tablet swiftly reaches the stomach. In the stomach, also known as the ventricle, the tablet enters the gastric juice. This fluid, produced by the gastric mucosa, is highly acidic and colorless. Some tablets are coated with an enteric coating to resist gastric acid. This coating not only provides protection for the gastric mucosa against irritative drug molecules, but also serves as a barrier.
But look, however, in this case we have a film-coated tablet, and it can be observed that the outer layer dissolves in response to the acid. Subsequently, the tablet progresses into the small intestine through the pylorus. The rate of gastric emptying can vary, which in turn affects the absorption time. Typically, the presence of food tends to slow down the emptying process. The small intestine plays a crucial role in the gastrointestinal system, as it is where the majority of drug molecules are absorbed. Upon reaching the small intestine, the tablet disintegrates and dissolves, releasing the drug molecules, which are then separated and prepared for absorption across the gastric mucosa.
Inside the small intestine, millions of villi, small finger-like projections, extend into the intestinal lumen. These villi greatly increase the internal surface area of the intestinal walls, providing a larger surface area for absorption.
And the absorption of most drug molecules occurs through passive diffusion along a concentration gradient, facilitated by the mucous membrane. However, a minority of drug molecules primarily rely on passive or active transport through specific transport molecules present in the intestinal cell membrane.
The extent of absorption is influenced by various characteristics of the drug molecules. For example, molecular weight plays a role, as small and lipid-soluble molecules are absorbed more rapidly. On the other hand, proteins are too large to cross membranes and thus must be administered through injection rather than orally.
The transit time in the intestines can also affect the absorption rate. In patients with diarrhea, the transit time is shortened, potentially leading to reduced absorption.
We have now entered the intestinal blood circulation. The drug has diffused across the epithelial cells that line the intestinal lumen, continuing its journey into the bloodstream through the portal venous system. Alongside red and white blood cells, proteins, and nutrients, the drug is transported with the bloodstream to the liver.
In the blood, many drugs bind to proteins, with albumin being the most common binding protein. However, there is also a portion of non-bound drug molecules known as the free fraction, which is the active form of the drug. We are now entering the liver, an organ responsible for detecting and eliminating exogenous compounds, including therapeutic drugs, as it perceives them as toxins.
The liver is composed of 50 to 100,000 small units called lobuli. Drug molecules enter the liver cells, known as hepatocytes, through small openings in the lobule walls. Within the hepatocytes, many drugs undergo transformation into compounds that can be easily eliminated from the body, either through urine or bile.
Some drugs are converted to active metabolites with pharmacological effects, while others generate inactive metabolites. Here, we can observe that the drug molecule undergoes a chemical modification, resulting in the attachment of a chemical group. This modification increases the water solubility of the molecule, preparing it for elimination through the urinary system.
This metabolism, before the drug enters the systemic circulation, is also referred to as first-pass metabolism. Some drug molecules bypass liver metabolism and enter systemic circulation unchanged. The proportion of drug molecules that remain unaltered in the bloodstream after absorption and first-pass metabolism is known as bioavailability. Bioavailability can range from 0 to 100%, with the latter indicating that all administered molecules enter systemic circulation.
The drug continues to travel through the bloodstream, following the path of the vena cava, and eventually reaches the right side of the heart. Then proceeds through the lungs before finally arriving at the left side of the heart.
From there, with each heartbeat, the drug molecules are propelled throughout the body, reaching their intended site of action.